Okay, and we are recording. So, hello everyone. Thank you for being here. My name is Cori Zuvia, as mentioned. I am a neuroscience major and I work in Dr. Prue Talbot's lab, toxicology lab. And today we will be looking at my research on the effect of passive cigarette smoke exposure on children's health. Particularly, we are looking at adisoprostane and metallothionein. Give me one moment. Can you guys see my screen?
We can't yet. I was going to let you
Okay,
know. Sorry about that.
there we go.
There we go.
Perfect. So just to reiterate, today we're looking at my research on the effect of passive cigarette smoke exposure on children's health. We are studying adisoprostane and metallothionein in these children. And so what exactly is passive smoke exposure? Passive smoke exposure is the exposure to cigarette smoke that isn't through the direct and active use of cigarettes themselves. So one type of passive smoke exposure is secondhand smoke, and it is defined by the CDC as the inhalation of smoke exhaled by a smoker, as well as the inhalation of smoke from the burning end of a cigarette. And so since we are looking at children, we can see in this image here that this child is being engulfed in a cloud of smoke and is unwillingly inhaling the smoke. Another type of passive smoke exposure is third-hand smoke, which is defined by the Mayo Clinic as tobacco residues that serve as environmental contaminants and settle onto surfaces like carpets, clothing, furniture, and even baby toys, which can then be inhaled, ingested, and dermally absorbed. And so, since we are looking at young children, it's important to note that children navigate their world through touch and through putting things in their mouth. And so, studying these young children and passive smoke exposure is important because it may give us an indication of the mode of exposure that these children will be exposed to. And something else to note here is this is the house of a smoker, and these are the residues on the walls. And these third-hand smoke residues don't simply wash away with normal household cleaning items, but instead can maintain in the environment for years. And so, why are we looking at passive smoke exposure? Well, previous research does show that cigarette smoking itself causes oxidative stress. And so we believe that this oxidative stress may also be shown in passive cigarette smoke exposure. And so one type of oxidative stress is adisoprostane, which is a prostaglandin that forms upon lipid peroxidation. So essentially, you just have a normal fatty acid here, which is a lipid, and when it is put under oxidative stress, in our form, passive smoke exposure, it will then be converted into this molecule adisoprostane, which is what we are looking at. And so, if we are changing the structure of our normal fatty acids in our body and converting them into adisoprostane, those fatty acid lipids won't be able to do their normal function in cell functioning and cell-to-cell signaling. And this conversion can ultimately result in diseases like cancer, inflammatory diseases, and cardiovascular diseases. Something else that we are looking at is metallothionein, which is a biomarker of metal exposure and reactive oxygen species, both which result from cigarette smoking, and we think passive cigarette smoke as well. And so here's the structure of metallothionein. It is a protein with these beta and alpha domains. So essentially, if there are metals or reactive oxygen species in your body, metallothionein will bind to it. So we should expect to see more metallothionein in those exposed to passive cigarette smoke. And so what do we already know? Actually, in our lab, one of our researchers, Dr. Sakamaki Chin, has found that these biomarkers of harm mentioned have been significantly elevated in active cigarette users. So Here, we are looking at those who use cigarettes directly. We have 8-isoprostane here and metallothionein here in our mean and standard deviation graphs. And there were 3 groups that Dr. Sakamaki-Ching looked at: a no-smoker group, a cigarette smoker group, and a e-cigarette group. And what they found was there are significantly elevated levels of these biomarkers of harm in the cigarette users and e-cigarette users compared to our non-smokers. So if we are seeing this trend in adults who are using cigarettes directly, what are the effects of children who are mere bystanders in this case? And so we wanted to look at that, and we hypothesized that children passively exposed to cigarette smoke, whether it's secondhand smoke alone, third-hand smoke alone, or in combination, we believe that this passive exposure in children will result in elevated levels of 8-isoprostane and metallothionein, specifically in urine. And so the way that we are testing this is through analysis of spot urine collected by Cincinnati Children's Hospital across 3 cigarette smoke exposure groups. So here we have a no exposure group, our control, abbreviated NEG, and then we will be comparing this to a third-hand smoke exposure group, TEG, which only has these third-hand smoke residues. And we will also be comparing our control to a mixed exposure group, MEG, which has a combination of secondhand smoke and third-hand smoke. And so it's important to note that our sample sizes are relatively equal across all of our groups, and this color scheme will be the same throughout the presentation. And so within those groups, we quantified the levels of 8-isoprostane and metallothionein by using a series of enzyme-linked immunosorbent assays, abbreviated ELISAs. So essentially what that is we have a 96 well plate, and the wells are coated with this capture antibody. And when we add in the sample, it can either have a low amount or a high amount of the biomarker, which will then bind to the capture antibody. We can then add in a detection antibody, which binds to the biomarker. And lastly, we add in the substrate, which binds to the detection antibody and causes a colorimetric reaction to occur. And from that color intensity, we can calculate the amount of 8-isoprostane and metallothionein in the children's urine. And so since we are working with urine, there are variables that can occur, such as what time the urine was collected or how much water an individual drank at that time of day. And so we account for this by using creatinine, which is a reliable protein for normalizing our biomarker concentrations. It's reliable because it's released from the body at a constant rate, and it's proportional to muscle mass, which does not change daily. So essentially what normalization is we take our biomarker, the metallothionein and 8-isoprostane, we divide it by creatinine, and now we have a normalized concentration to make our data more accurate and reliable. And so looking first at that data, we have our creatinine concentrations. We have a mean and standard deviation plot with our x-axis containing our no exposure group, our third-hand smoke exposure group, and our mixed exposure group. And we have creatinine on the y-axis. And what we see is that there was no significant difference across the exposure groups. And this is great. It's creatinine, so we're using it as a normalization biomarker, and we don't want it to affect our oxidative biomarker data. And so now getting into that data, the exciting part, we first have 8-isoprostane, which, if you recall, is that lipid peroxidation. And what we see in our mean and standard deviation plot is first, there is no significant difference between our no exposure control and our third-hand smoke exposure group for our 8-isoprostane. But when we look at our mixed exposure group, we see that it is significantly elevated in 8-isoprostane compared to our no exposure group. And while we do have some outliers here, we can ultimately see that the means for the MEG, mixed exposure, is significantly higher than the no exposure. And we actually see a similar trend when we look at metallothionein. Excuse me. So metallothionein is indicative of metal and reactive oxygen species exposure. And what we see once again is that our no exposure group and our third-hand smoke exposure group are not significantly different. Yet, our mixed exposure group is once again significantly elevated in metallothionein compared to our control. And so, what is this telling us? Well, we see that the third-hand smoke exposure group isn't seeming to have an effect. Yet our mixed exposure group here, which contains secondhand smoke and thirdhand smoke, gives us another interpretation. So we can say either secondhand smoke alone is causing an increase in the biomarkers, or maybe it's this combined synergistic effect of secondhand and thirdhand smoke that's causing this trend in an increase in biomarkers. And so, although it is hard to control for only having a secondhand smoke group in human studies, this mixed exposure group is important because, and most relevant because children living in homes of smokers will inevitably be exposed to both secondhand and thirdhand smoke. And not only this, but we can also interpret our metallothionein data in a different way. So we know that metallothionein is indicative of metal exposure and reactive oxygen species. So it can either be those individual factors or a combination of both that is causing this increase in metallothionein in our mixed exposure. And so, in summary, our data is saying that the TEG children did not display significantly elevated levels of metallothionein or adisoprostane compared to our no exposure group. Yet, the MEG children did have these elevated levels of biomarkers compared to our control. And so, why is that important? So, our data is appearing to conclude that third-hand smoke alone is having no effect. Although in the toxicology world, there is actually some data in mice and in vitro studies that is showing that third-hand smoke is causing increased molecules that are associated with cancer, cardiovascular disease, and lung disease. And so, while in our human study right now, we aren't seeing that third-hand smoke is having an effect, we did have maybe a small sample size, and more research definitely needs to be looking into humans and the effect of third-hand smoke on humans. But what we can say even more so is that this analysis of the mixed exposure group, the secondhand and thirdhand smoke, is extremely important because, as I mentioned before, children will inevitably be living in households of who live in households of active smokers will inevitably be exposed to both secondhand and thirdhand smoke. And this finding is translational to toxicology because not a lot of research has looked at children and passive smoke exposure. And not only this, our data may be a gateway if we're looking at the synergistic effects of secondhand smoke and thirdhand smoke, as opposed to treating them as separate entities, as a lot of passive smoke exposure studies do. And so, although we would consider our research complete right now, some future directions for others to take may be to look at metal concentrations in children's urine and see if metals are positively correlated with metallothionein. So, right, we focused on reactive oxygen species in our studies, but maybe metal is also having a profound effect and may lead to metal toxicity that we haven't studied in the children. Something else to consider is to develop more, to analyze data based on gender and age. So collecting Cincinnati Children's Hospital can perhaps collect data that has more equal sample sizes of gender and age. And we can then look if into gender is more prone to oxidation or does age suggest method of exposure, right? Younger children are more likely to put things in their mouth. So are the younger children more susceptible to exposure? We don't know that yet, but that would be great to look at. And lastly, another thing for future research would be to analyze other markers of harm and really expand the other effects such as DNA damage, protein damage. But I just wanted to thank my lab. Thank you to Dr. Prue Talbot, and thank you to Mark for giving me the tools to succeed in research and the NIH for funding my research. So thank you.
Great job, Cori.